And I'm going to turn the remainder of the time over to Warren Wagner, research botanist and curator of botany at the Smithsonian National Museum of Natural History. Thanks so much, Warren.
Thanks, Lauren. I'm going to just make a few sort of general sorts of comments because this session was kind of the introduction to getting into many of the details that have sort of been laid out and a lot of the questions raised already in the remaining sessions of the conference. But I hope you found the presentations that we put together for the opening session interesting and informative. And we know it was a lot of information across a broad range of topics, but our goal was to introduce the workshop series, and especially with the potential of integrating new molecular tools to generate additional data to be more informative to go along with our own ongoing studies of Hawaiian lineages using more traditional approaches of comparative biology. Adding molecular data to these studies of lineage of species, populations within species can greatly increase our understanding of them, as we've already seen, especially when coupled with other information, as Steve laid out to some extent in the first one, on things like breeding system, pollination, biotic pollinator biology, and pollinator distributions. We know very little about a lot of those. We didn't really get into that, but it's a big lacuna for Hawaiian lineage studies, as well as study of additional populations from what we've been able to do so far, even though many of them are reducing rapidly. Allowing additional kinds of molecular tools to enter the system, it can provide valuable data for making better management decisions and designing initiatives to conserve rare species. Although sometimes they don't work, but hopefully as we learn more and try other different things, such as maybe some test plots on recovery, plantings not in the ground, but maybe testing areas with plants in pots because we know so little about distribution of pollinators, for example. Adding this kind of information will, of course, improve our classifications as well as provide valuable data in making difficult management decisions. The NSF grant that has made this workshop possible has the focus of using high throughput DNA sequence generated by the project on the endemic genus Cheetia, which actually has already had a morphological analysis and two Sanger sequence analysis, one using multiple genes, one using fewer. And we have high hopes already seen from the preliminary data that these new methods will greatly help our understanding, especially of lineages that are diversifying in such a young framework of a few million years or less. And this understanding can aid us in better designing our conservation reintroduction efforts. We also hope that by integration of these approaches and the expertise demonstrated by the team we put together, that this is a good model for a way to go that is of possible use across many of the poorly understood Hawaiian and other island lineages. Of course, the foundation for all of our classifications is morphology. You know, to look at patterns of variation in species and search for patterns of correlated characters and discontinuities among populations. But research, including systematics, is an iterative process of building, refining, and improving classifications. I know it irritates people to always have new classifications coming around, but that's the way it is as new data and new studies and new understanding come to pass. And these can help us refine the classifications and also understand the evolution of the group and interactions among species, which we also know relatively little about and is based primarily on morphological intermediacy in certain populations and individuals. So in addition to that, it's beyond the basic morphological studies, which has been the hallmark for a couple of centuries. As new kinds of studies are developed, it's really great to be able to add these to increase our understanding, to not only understand the pattern of variation in characters, but understand their function, and also add areas from characters such as micromorphology, which have not been added before, crossing information, breeding systems, and pollination, as well as adding kinds of molecular data that we've been talking about from multiple parts of the genome as appropriate. So, and each of these additions gives rise to new ideas, which in turn feed back into the data collection and interpretation. Integration of all available data sources really help refine species levels, and that that it's an iterative process, and integration are two really key aspects of this. For example, it's just hearing some of the talks made me think of one thing, mentioning the Hawaii flora. When that was started, many of you probably already know, but others may not, there hadn't been one synthesizing mostly morphological information for 100 years. And we didn't have a lot of time, so we took a quick snapshot using the soundest principles as we could, but primarily for morphology and not even as much field work as would have been desired, mostly from herbarium specimens. But that snapshot sets the stage, as mentioned, for additional integration of study of additional populations, new approaches, and techniques. So, for example, the Sheedia work, which the current grant was originally 2 genera in the Hawaii Flora Manual, based on the very different morphology of what seemed to be bird-pollinated and biotic or wind-pollinated species. There are 26 species. And then when Steve and Ann entered and we formed collaborations with each other and many other people, and the many studies that have emerged, it increased from 26 to now, with the species just being worked on now, 35 species. That's quite an increase in understanding how the partitioning of variation and the function of these different groups of populations are. There are many, many other lineages in Hawaii where we don't have that kind of information, and we greatly need it, especially when it comes to conserving and doing recovery plans. So in closing, I just wanted to say that trying to do so many different kinds of approaches in a group is really hard for one person or a small research team. But as we were trying to demonstrate here, far easier and faster, and possibly more in depth if they're done collaboratively. So, and another reason for collaboration is it can ensure that researchers versed in various approaches understand the methods and especially their strengths and weaknesses, appropriate application, and more experience at interpreting the results. Teams of researchers can better design appropriate and efficient projects. And when academic studies of species and their evolution and their biology join forces with conservation biologists and field biologists, we can develop the best possible conservation initiatives for the many rare plants and lineages in the Hawaiian flora. So with those comments, I'd like to turn it back over to Lauren for final comments and info on the upcoming sessions. Thank you.
Thanks so much, Warren. That was really great to get us started today for our journey on the remainder of the series. I'll have a lot more details to share about that format with all of you tomorrow. We'll be starting 1 hour later at 9 a.m. Hawaii Standard Time, and it's the same link. Please reach out to me if you have any questions in the meantime. And if you have any questions about that you thought of today for any of the presentations, we'll be there tomorrow and we'll have time to address those questions tomorrow as well.